Thanks. It's a real pleasure for many reasons to be here in San Diego. I'll just say that the temperature in London is 32 degrees and it was raining this morning. So that is one reason that it's nice to escape. And I'd like to thank the Shiley Foundation for really supporting my visit here. This is really an honor and thank you very much. I try and come to, as you can imagine, I try and come to San Diego about this time every year to the Salk Foundation and to the Salk Institute just next door. It's always a pleasure to come. I've got a lot of friends here, so it's really nice. But I'm going to start actually by talking about a real bit of history about San Diego. I'd just like to take this opportunity to thank, sadly posthumously, of course, Leon Fahl. I was recruited to the University of South Florida in 1992 from London, and I didn't, well, perhaps nobody understands the NIH grant system, but as an Englishman, I certainly did not understand the US funding system. And I was hired in Tampa in 1992. And by early 1993, my lab, which at that time included Mike Hutton and Karen Duff, who are now fairly, well, actually, in fact, Karen Duff's now my boss. So, you know, I have to say nice things about her. I would do anyway, but, you know, my lab was me, Mike Hutton and Karen Duff and a Ph.D. students. And we were running out of money. At one point, we were 3 weeks from closing down in 1993. And I'd never met Leon Fahl at that time, but he got wind of our problems and he organized that one of his donors would send us $75,000. And that was in 1992 or 1993. I don't know how much that is today, but maybe that's equivalent to about $300,000 or $400,000. And that actually is what bailed us out. The lab would have had to close except for Leon Fahl. I'd never met him. And a year or so later, I did meet him and went up to him at a scientific meeting and, well, attempted to say thank you, and he waved me away. This was just the right thing to do, was his attitude. He didn't want personal thanks. I never had to do anything in recompense. It was just straightforwardly a donation to get my lab over the bump. And so, you know, I think that's quite remarkable. And I think that's really quite remarkable. And that sort of generosity is rare. So I'd just like to say thanks to San Diego. So it's a pleasure to be able to finally to say thank you. Okay, so I'm going to talk nearly all about Alzheimer's disease, nearly entirely about Alzheimer's disease. In the first few slides, I'm going to talk about some of the other diseases too. I'll just start with the pathologies. Here's the pathology of Alzheimer's disease with the plaques, middle center, the tangles, bottom left, the amyloid angiopathy, which of course we all ignored until recently, bottom right, and then the gross pathology at the top left, showing the enlarged ventricles, top right, the bottom of that showing the shrunken hippocampus. This is the pathology. Of course, you know, seeing this pathology, it wasn't clear which part of that pathology came first. And when we started, you know, when you go to Alzheimer meetings, I'm quite old, well, I say unfortunately, the alternative is less good, clearly. When we started, I started going to Alzheimer meetings in the late 70s, very late 70s, early 80s. At that time, Alzheimer meetings would have about 40 or 50 people at them. Bob Terry, of course, was one of the stalwarts at that time. And for all the 40 or 50 people, there were 10 or 20 theories about how the disease started. People thought it was slow viruses. People thought it was accelerated aging. People thought it was aluminium saucepans, all sorts of ideas. And in 1983, Gusella had shown that the Huntington's gene was on chromosome 4. And I was working on the pathology of the disease. I'm a biochemist, but I was working on the pathology of disease. And here you can see the pathology. And what occurred to me was that we were looking at the end of disease. And here was Jim Gusella's approach, which was going to find how the disease started. So I thought, how cool is this? If you could understand how the disease starts and we can see how it ends, you can basically referee between the different theories. So I always saw genetics as a way, as the referee about how disease started, rather than, between all these competing theories. So after Gusella's paper came out, I was fortunate enough, even though I was a biochemical pathologist, to be hired by a genetics department to do their teaching of biochemistry, fortunately for me. And so the quid pro quo was I did the medical school biochemistry teaching. I can tell you all about the citric acid cycle and glycolysis, but their side was they taught me molecular genetics, which for me was really, it was just great. And I had a great head of department. Anyway, that's the pathology of Alzheimer's disease. This is the pathology of Parkinson's disease. Top right, normal control substantia nigra, showing the black line of the dopaminergic cells. Top left is the faded nigra, because you've lost a lot of those cells. And bottom right is the synuclein stained Lewy body. Here it's worth mentioning that really synuclein staining in a way started here with Tsuno Saito developing antibodies to synuclein. So some of that history is a local history here too. So anyway, that's the pathology of Parkinson's disease, and this is the pathology of the tangle diseases. There's a number of tangle diseases. This is the most common of them, progressive supranuclear palsy. This has tau pathology in the, for example, the dentate gyrus. Bottom right, you can see the pathology, the tangle pathology. They're made up of 4 repeat tau. Now, why have I shown all this? Perhaps the simplest genetic finding that has been made in all of these diseases are gene duplications. We showed, top left, synuclein gene duplications cause Parkinson's disease. Dominic Campion, bottom left, showed that APP, amyloid gene duplications, were a very rare cause of Alzheimer's disease. And his group also, top right, showed that tau gene duplications were a rare cause of tangle disease. So these are extremely rare. I know that, for example, in the UK, we have 7 families with APP gene duplications. So in terms of epidemiology, they're trivial. But in terms of mechanism, they're extremely important, because what they tell us is that these proteins, all 3 of them, are very close to what one might call their crystallization threshold. You make just a bit too much, 50% too much, and you're going to get sick. In all of the cases I've mentioned, you get sick in the 50s. So all of these cases are close to their deposition threshold. I'm making that point, but from a simple genetic perspective, but the same point has been made by Chris Dobson's group. Chris died about 7 or 8 years ago now, but his work continues with Michaela Vendruscola in Cambridge. he showed the same thing from a protein chemistry point of view. He showed that all the 3 proteins that are deposited are close to their deposition threshold. Now, these mutations which cause simple overexpression are very rare. Mutations we know about in the same genes, point mutations, make the deposition more likely. For example, APP mutations, which either cause more amyloid production or cause the production of a less soluble amyloid protein, also cause disease. But all of those together are rare. What's important, though, is the subheading. The implication is that if you just overexpress the protein, you're going to get sick. Then if your clearance mechanisms are slightly deficient, you're going to get to the same outcome. And that turns out to be, I think, very important. I'm not going to talk about Parkinson's and tangle diseases anymore, but just to say that the evidence that we have is that synuclein is cleared largely through the lysosome. So lysosome, if you like, deficiencies underpin a lot of Parkinson's disease. And we think, though the data is less good in this, we think that the ubiquitin proteasome is important in clearing tau. That the data, for reasons I won't go into, is less certain about that. But as a general rule, then, if you've got a problem in clearance, you're going to get into a problem in deposition. Now, I get both the credit and the blame for the amyloid hypothesis. In fact, I don't really think I deserve either, because the first statement of the amyloid hypothesis came from George Glenner. Actually, I'd forgotten it until I showed this slide. George also is a UCSD alumnus. I'd forgotten that. Yeah, this is also, I think this slide is from before he came here, but he was also a UCSD alumnus. And he wrote about Down syndrome. The first thing he got was a sequence of the amyloid peptide from the congophilic angiopathy earlier in 1984. And then knowing that Down syndrome individuals always got Alzheimer pathology, got the same sequence from a Downs individual. And he said, and I'm trying to read this, my eyesight unfortunately is failing. I'll summarize what he said because I can't read it anymore. But what he said was he thinks that the amyloid gene must be on chromosome 21, and that individuals with Down syndrome get the disease because they've got an extra copy of that chromosome. And he turned out to be right on both. I love predictions which are before the fact. You get a lot of people who make what one might politely call post hoc predictions. I'll talk about some of those later. So anyway, that's the first almost explicit amyloid hypothesis. He sadly died in the, I think, early 2000s. But the same work was really published by Colin Masters, who's still very active in Melbourne. And he was involved in the cloning of the amyloid gene with Konrad Bayreuther, and he too clearly thought that amyloid deposition was how it started. So this is how we came to it. This is the family that cracked it for us. This is a family from the centre of the UK, a town called Nottingham, kind of equivalent to, let's say, Cleveland here in the US. What you're looking at here is a grandfather with Alzheimer's disease. His 2 sons had Alzheimer's disease. His 2 sons married 2 sisters. And that's not important for this, but it's the fact, so I show it. And then the next generation, 10 children, 5 of whom get the disease, and a double cousins, 1 of the 3 get Alzheimer's disease. And you're looking at the top half of chromosome 21, and the 9th individual has got the very top of the chromosome. the 11th individual has got the bottom of that chromosomal segment, and that puts the disease gene in the middle of there, and you can see the APP gene is in that middle. So that's how we got there. All sounds very simple. This slide took 5 of us 6 years to do. At that time, the human genome wasn't sequenced. We didn't have PCR. We got PCR just, I know I sound like a real old fart when I say this. PCR had not been invented. We didn't know the order of genes and so on. But we made other mistakes, but one crucial mistake. And that is that we thought that, like Huntington's disease, there would only be one Alzheimer gene. That was the mistake we made. So we pooled all of our families together and had done joint analyses. And that had misled us. And it was that it was finally twigging that we should have analyzed the families one at a time that was key to the analysis I've just discussed. The other thing I also should mention, which was also key, was that there was, there still is, a disease called Dutch congophilic angiopathy, which is an amyloid deposition, but as the name suggests, the amyloid is deposited only along the blood vessels, and with Blas Frangioni on the one hand and Christina van Broekhoven, who we were collaborating on the other, showed that amyloid mutations cause that disease. So we had 2 bits of data, if you like. Our analyses pooling all the families were wrong, and we had this precedent of Dutch congophilic angiopathy showing that amyloid depositions caused at least part of the same pathology as Alzheimer's disease. The paper at the top left there, that's the official version of cloning of the amyloid gene and the mutations. The paper at the bottom there is, when I lived here in the States, I used to watch a terrible program which was called, I think, Hollywood Behind the Headlines. And it would always be a story of usually a child star who had got into drugs and his life had gone off the rails. And then at the end of the episode, they'd gone clean and they were now recovering. I was a sucker for watching that every week. Well, I was asked to write, if you like, Alzheimer's disease, the cloning of the amyloid gene behind the headlines. And I won't say that drugs were involved, but let me just say that the story at the bottom left is more honest in terms of the mistakes that were made. And it might be more interesting to read, if you like, if you're a sucker for that sort of thing. Okay, so this is when we wrote the amyloid hypothesis. My papers are on the right-hand side. Dennis Selko reached the same conclusion at the same time, and his paper is on the left, and that's usually taken as the starting gun for the amyloid hypothesis. Then when we had in our family series of about 30 families collected by Dr. Martin Rosser, who's still my colleague at Queen Square, of those 30 families, only 3 of them had amyloid mutations. So obviously there was at least one other gene involved. And so then there was a race to, in those, well, I'll talk about that in a second. There was a race to find the other genes. I love those days. In those days, it was nature read in tooth and claw. The different genetics groups were absolutely ruthless competitors with each other. And we would listen for any whispers about each other, and we'd each collect our own families, and so on and so forth. So that was how we operated. We were read in tooth and claw. What happened later was we all had to learn how to play nicely in the sandpit as we moved from the single kindred era to era when we pooled all our data. So we had to go from ruthless carnivores to placid vegetarians almost overnight after when we ran out of families. Anyway, that's by the way. So the nature red in tooth and claw era, I think the person who really sorted that, well, did the most there was Peter Hislop, who cloned first presenilin 1 and then presenilin 2, and Jerry Schellenberg had identified a locus for presenilin 2 and found mutations in presenilin 2. Incidentally, we have never in the UK found a single family with a presenilin 2 mutation. It's kind of strange. I don't know why that is. Anyway, so presenilins really fitted with the amyloid hypothesis. The structure of presenilin is now known, beautifully worked out by Shaw-Sherez at the LMB. But this in blue is the presenilin protein, 9 transmembrane domains, and crucially, the amyloid precursor protein is drawn in its active site, and all the residues in red are residues at which we and others have found pathogenic mutations. And in the transmembrane domain 6 and in the transmembrane domain 7, you'll see a turquoise residue. That turquoise residue is an aspartate, and that aspartate is the active site of the cleavage, which cuts across in the amyloid precursor protein. So this, of course, fits with the amyloid hypothesis, because here you're looking at the amyloid protein as, if you like, the substrate for presenilin gamma secretase. And the key work here was really done by Steve Youngkin, who was at Mayo Clinic. Well, I was going to say with me. In fact, I was at the Mayo Clinic with him. He was my boss at that time. And the presenilin showing that all the mutations in presenilin and several of the mutations in APP affect the cleavage position of the presenilin gamma secretase. The work on the precise mechanism of this has been done by Bart de Strooper and Lucia Chavez Gutierrez. Their papers on the precise mechanisms are really beautiful papers which show the mechanism. I'm not going to go into it. So, at the same time as we were identifying families with Alzheimer's disease, of course, we were also picking up, often by accident, families with other dementias which were referred to us. And we had collected, our group had collected in particular, one very large family from Northern Australia with dementia, which turned out late, which was at that time we showed was chromosome 17 linked. And after a period, we finally worked out that, in fact, the tau protein was, sorry, I didn't say this, and it's crucial. These families had turned out to have tau mutations, and their pathology was only tau pathology. The work showing the pathology was beautiful work done by Maria Spillantini, really important work. And she and Jerry Schellenberg and we all found tau mutations. Again, after making mistakes. I'll explain the mistakes because the mistakes are interesting too. And that is that one set of families showing linkage to chromosome 17 turned out to have tau mutations. Just about 2 megabases away on the chromosome 17 is the progranulin gene. So there were another series of families which had mutations in progranulin. And again, we made a mistake lumping all the families together. And in fact, I think it's okay now to say this. The bottom right paper is Jerry Schellenberg's paper. Jerry Schellenberg's paper had been accepted at the Annals of Neurology, and at the proof stage, the title of this paper was changed from tau is not a candidate gene to tau is a candidate gene. So that was a pretty substantial change, which passed the editorial board. And they did it because what had happened is they had mixed the families up, the progranulin families with the tau families, and had realized that the majority of families did not have tau mutations. So on that basis, they had ruled it out. And so they heard anyway, it's good to get this scuttlebutt. Anyway, that's what happened. At least that's my story. So why have I told you this story about tau mutations? Well, because this allowed us to make mice with tangle pathology. And we now had, and others, Karen Chow and the Athena group, have made mice with plaque pathology. So we could now cross amyloid mice with tangle mice. And so the advantage of doing that, of course, you get plaques and tangles, but you can also start to see an interaction between them. And when we crossed the amyloid mice with the tau mice, what happened was the amyloid pathology stayed the same, but the amount of tangle pathology went through the roof. That's shown best in those middle histograms there. And so that puts amyloid upstream of tau and plaques upstream of tangles. And that allowed us to draw this incredibly simple-minded amyloid hypothesis diagram. I still think that this diagram has some truth. Look at the year, September 1998. But also look at the question marks, and those question marks are still here. And so, you know, one of the things about science is if a question mark is still there 28 years later, maybe it wasn't such a good question. That's really, and that's, I think, the point. And this diagram, although it has some truth to it, I think is wrong and misleading in a series of related ways. The first is, there's a line there, the vertical line with the question mark. It implies that amyloid outside neurons causes tangle in neurons. The only cell in this is a neuron. That is the only cell in that diagram. And that is one way it's wrong. The other important way it's being wrong is I had always thought that what we would do as geneticists would delineate something like, because I'm a biochemist, something like glycolysis. I thought we were going to be looking for a pathway like glycolysis and be able to dissect the pathology like glycolysis. Of course, glycolysis, there was if you change the glucose concentration, your concentration of TCA intermediates goes up. Actually, it goes up in much less than a second. These biochemical pathways really move. What we're talking about in this diagram is a process we now realize takes 20 or 30 years. Actually, again, this was first shown indirectly by my colleague in Manchester, David Mann, but has been beautifully illustrated by those, what are they called? I can't remember what the curves are, which are always show the, Doug, what question? The Jack curves. Thank you. My hippocampus is going down a bit. So there, the Jack, those be beautifully shown by the Jack curves. It's 30 years. That is not a biochemical pathway like glycolysis. Now, when we go around debating the amyloid hypothesis, people would say, you know, I believe the amyloid hypothesis, like it was a religious doctrine, but I believe the amyloid hypothesis, but, only for Down syndrome and the amyloid mutations, and maybe I believe it for the presenilin mutations. But in late onset Alzheimer's disease, you have no idea what's going on, and it's not right to extrapolate from these very rare cases to the disease in general. That extrapolation is really unsubstantiated. And that, of course, is a fair point. So the question becomes, what underlies late onset Alzheimer's disease? Now, we couldn't do anything. We geneticists couldn't do anything. And if you look at the literature, ApoE was found by Alan Roses in the 1990s. But actually, the presenilins were found, I can't remember what year, but let's say the early 1990s. And we geneticists didn't find anything apart from ApoE till 2007. We had 15 years without any findings. And what we would do in those 15 years is test one gene after another for association. And we always failed to come up with any genes. And it was only with the invention of genome-wide association studies that we broke the deadlock. And so fortunately, the NIH kept funding us for those 15 years, even though we didn't have anything to show for it. So it was only with genome-wide associations that we finally began. And the power of genome-wide associations is that they survey the whole genome. They are completely statistically sound. You're doing about 500,000 chi-squared tests, so you have to set your Bonferroni correction really high. So you set the Bonferroni correction, which on this diagram is the horizontal red line. It's at about 10 to the 7. And if you exceed that with one of the SNPs, you're allowed to declare it. It's robust, it's statistically sound, and it's a technology which works. Of course, actually, again, these are on Illumina arrays, and I'm forgetting how much San Diego comes into this story. So, this is a San Diego company. I hope they're listening, and I hope they give you some money. So anyway, and now what we had expected was to find a lot of APP genes, APP-related genes, and there's a couple of APP genes. There's one of the alpha secretases is over here. Again, my eyesight isn't quite good enough to call it, but it's one of the ADAM proteins here. But the majority of these were really a surprise to all of us in the field. They were not obviously amyloid related. Notice that obviously is in italics, because they turned out indirectly to be amyloid related. This point was first made by Leslie Jones in her paper on the left-hand side. She pointed out that the majority of these genes were microglial, and the majority of them, she says, cholesterol related, but actually I would say lipid related. How does that fit? Well, I'll come back to that in a second. We had started to sequence Alzheimer cases with the new technology of, at that time, exome sequencing. And we had concentrated, one of the first genes we looked at was a gene called TREM2. We had looked at TREM2 because with colleagues in Turkey, we had found TREM2 mutations caused a disease now called Nasu-Hakola disease, a leukodystrophy. And so we were looking, and when you find a gene, what you do is you go through your own database, first of all. Have I ever seen mutations in this gene? And nowadays, especially, you go through the public databases and say, has anybody else found mutations in that gene? So you do look in your own database and so on. And with our colleagues at Mayo and elsewhere, we looked for the mutations we had found in the Turkish Nasu-Hakola disease cases, and to our surprise, found the same mutation in English and North American Alzheimer cases. In Nasu-Hakola disease, they were homozygous. In Alzheimer's disease, they were heterozygous. So the same gene which, when homozygous, gave you this rare white matter disease, when heterozygous, increase your risk of Alzheimer's disease by about 3-fold. We were also a different part of the lab, entirely separate, was looking at mice with amyloid deposition. And we were doing a simple experiment. What we were doing was looking at mice which were depositing amyloid and saying which genes go up in expression when amyloid is deposited. And to our surprise, TREM2 is the gene which goes up the most in response to amyloid deposition. So we had two completely independent findings. TREM2 is a risk gene, and TREM2 is a gene which responds, the gene which responds to most, to, you know, you don't get, that's a coincidence. Well, when you get coincidences like that, you think, I have to look and see if I can find more. And so that's what we did, and this was done by my colleague Dervis Saleh. And this is the same data on the right-hand side, only more with more genes shown. And what Dervis showed is that not only was TREM2 one of these, the gene, TREM2 is right in the middle here, circled in with the red ellipse, but the others circled with the red ellipse are also Alzheimer risk genes. So this is saying that genetic variability in genes which respond to amyloid are risk genes, are risks for Alzheimer's disease. And so could we exploit that? You know, I said earlier on that what we were battling against in genome-wide associations was Bonferroni. We were battling that damn Italian statistician who made us set the p-value at 10 to the minus 7. If we could say, no, we don't want to set the p-value at 10 to the minus 7, we only want to set the p-value for those genes which respond to amyloid, which is about 10 to the minus 4 or 5, and then we can declare those genes. And that's what we did in the paper on the left there, and all the ones in blue are genes which showed statistical association with Alzheimer's disease, but didn't meet the strict Bonferroni correction, only met the corrected Bonferroni correction that I've just mentioned. And I won't talk about these genes, except I'm going to mention one of the genes later on, which is the gene on this slide. It's OAS1 in humans, or OAS. I can't read, again, my eyesight, God, I can't read it. So it's OAS though. OAS is a gene which we declared. And the reason it's interesting is because it's part of the interferon response system. That's why it turns out to be of interest. I'll come back to it later. So how can we make sense of all of this? And this is my suggestion. And I was really influenced by these papers and Leslie Jones' paper, which I mentioned before. Leslie, who had said lipid metabolism was what was important. The paper from Marco Colonna had shown that TREM2 was actually a phospholipid receptor, and there was work in the literature saying that ABCA7 was a phospholipid transporter. Now, when you see phospholipids, what do you think of? You think of cell membranes. And this was, for me, what underpinned my suggestion about what was going on. This is how amyloid metabolism is drawn, typically. And you can see how it's drawn typically. You'll see this everywhere in the literature. And you can see there that on the left-hand side, that's going to alpha secretase, and on the right-hand side, that's beta secretase, and then presenilin gamma secretase cutting at the C-terminal fragment. And then amyloid is shown going off into the extracellular space. This is shown everywhere. I've seen this diagram 1,000 times. I think it's wrong in a very simple way, and that is that amyloid, anybody who's ever worked with amyloid, for example, the lab next door to me is Henrik Zetterberg's lab. They work on, they have to be incredibly careful to avoid lipophilic surface. Amyloid, A-beta 40, but certainly A-beta 42, is extremely lipophilic. It touches a lipid surface and it sticks. So that's why you can't do, you have to choose your Eppendorfs very carefully if you're going to do any measurements of especially A-beta 42. And yet we draw A-beta being produced in the membrane and going off into the extracellular space. I don't think that's what happens. I think that what happens is that amyloid deposition begins in the membrane, and amyloid is produced in the membrane. The enzyme cuts in the membrane. Why should it float off when it's so lipophilic? One thing I should say is that I'm often asked which membrane it is, and I don't know. And so please don't ask me, because you won't get an intelligent answer beyond that. I guess that is an intelligent answer. So I think that what happens is that amyloid deposition begins in the membrane, and it disrupts the membrane domain, and that pulls in both ApoE and microglia, and that's what's going on. That's why microglia are involved, because they're dealing with this chronic membrane, I'm going to call it a membrane stressor of transmembrane domains disrupting membranes and bringing the microglia and ApoE to clear things. So I would say these are amyloid. This is how you bring together the amyloid, all the genes producing amyloid in early onset disease, and the genes involved in late onset disease being involved in clearance. And this is diagrammatically illustrated here. And what I've drawn here is in the red horizontal line, that's people with APP gene duplications, or perhaps people with presenilin and APP mutations. And the diagonal blue line is the capacity for removal. And when the capacity for removal is overwhelmed by the amount of production, that's when you start to hit trouble. And that's why those individuals with presenilin and APP mutations have an earlier onset, and those in the general population have a later onset, and the variability is illustrated too. So that's how I think you bring together early and late onset disease. Now, we had been, with Valentina Escott-Price, we had been concentrating on doing GWAS of path-confirmed Alzheimer's disease. And the reason we had done this is, I'd worked on the pathology of the disease. I realized that the clinical series were not, in fact, we worked out later, the clinical series as collected by all the UK and US centers was only about 70% clinically accurate. And so I thought that this was a mistake to get larger and larger GWAS, because actually, of course, as you make the GWAS larger, your quality control goes down as well. And that was illustrated to me by the fact that progranulin started to show up as a GWAS hit. Maybe it is, maybe it isn't, but as for the dementia series. So we wanted to do a path series. Of course, the number of path cases is very small. The GWAS I showed you before had 80,000 clinical cases. We could only manage about 2,000 path-confirmed cases. And so we decided to go to the ADNI data set. And my view at that time was they've got amyloid deposition data, so that will be the same as a path-confirmed. So getting the amyloid deposit case, the amyloid positive cases from ADNI would be a way of boosting our path confirmed cases, I thought. But what Valentina realized when she looked at these ADNI series was that they were different, and that the risk for ADNI amyloid positivity, and it was true in other series too, was almost entirely ApoE dependent. And we had done the same. Andre Altman and Mariam Shoai did the same, essentially the same analyses. And it was only dementia in the context of amyloid deposition, which depended upon microglia. So we are, if you like, dissecting Alzheimer's disease into at least two stages: amyloid deposition, ApoE, dementia in that context involving the microglial response. And that was done experimentally by this group here, who showed that if you did the experiment that we had done, amyloid plus tau mice, if you did that experiment, only you paralyze your microglia response, the tangled response was bigger. And so they too dissected the process into at least two stages. So that's where we've been up to in terms of risk. Now what we've been interested in, and this is really preliminary data, is we've been trying to do genetics of progression. In a sense, I've already illustrated part of progression by saying the microglial response is important. But what drives progression to dementia in the context of the microglial response? And to do that, we have been trying, well, finally successfully collaborating with drug companies, the people who have got the best longitudinal data together with amyloid and with good diagnostic criteria based on either PET scans or CSF, or more recently blood biomarkers. We've been working with drug companies, and we've been doing a GWAS of progression with the drug companies together with the ADNI data, which I've mentioned, and ABLE, which is the study basically headed up by Colin Masters, who I've mentioned, and others in Australia. And so we've been doing a GWAS. I have to say that collaborating, getting drug companies to pool data has been a lesson. Each drug company you contact, the scientists think it's a great idea. And then they say, I just need to put you in touch with legal. And honestly, that is, well, it's not quite the kiss of death, but it's certainly a wounding when you are put in touch with legal. Of course, I understand. And every drug company starts by saying, that's fine, we can collaborate with the other drug companies, but we need to have first right of refusal. And since every drug company starts like that, and your own legal department doesn't let you promise that more than once, unfortunately, it takes quite a time to bring everybody onto the same page. Anyway, there we are. That's where we're up to. We're now The first of our data, which is the ADNI-ABLE data, is now online. The ADNI plus ABLE data is available. I think it's on medRxiv, might be on bioRxiv. We're just starting. We have just got permission from the other 5 drug companies, from the 5 drug companies, to use their data, and we are just doing the meta-analyses. Now, these are the genes that have come out of the first GWAS. I think this is might be a slightly earlier version of this diagram, is on the medRxiv paper. The first thing to notice is that ApoE is not a hit. ApoE unambiguously is not a hit. It does not have a significant, it might have a tiny effect, but a significant effect of rate of progression if you define the cases by having amyloid deposition at the start of their inclusion. If you have amyloid deposition only at the end of the inclusion, then of course some people convert during that period and you do see ApoE. That's a very important distinction. What shows up? Well, it's underpowered. You can see that horrible red dotted line there. That's the Bonferroni correction. And you can see we don't meet it. What comes up though? Well, the first gene that comes up, and there are others in the same pathway, is the interferon response, OAS. So the interferon response comes up. I think we can be reasonably confident because some of these other hits are also interferon related, even though they reach genome-wide significance. So this is the ApoE zoom showing no effect of ApoE. Now let's talk about where we're up to in terms of treatment. This was the first amyloid antibody from Dale Schenk. Really caught, I think, everybody by surprise. I've tried to find out why they did this crazy experiment. It's obviously an experiment which is never going to work. And like all of experiments which are never going to work, of course, it surprises you when it does. And actually, I know the reason. I think I know the reason. And that is what they just had Joe Rogers and Pat McGeer to lecture to them at Athena. And what they were trying to do was, by pumping up the immune system in the context of amyloid deposition, what they were trying to do is to pump up the immune system to remove amyloid. That's what they were, to cause tangle formation. They were trying to say, if we pump up the immune system, can we get the brains to make tangles? And they just had Joe Rogers and Pat McGeer through to give them lectures, and I think that's what prompted them. So they were trying to make the pathology worse, and then they looked and the amyloid had gone. So there we go. Now I think, certainly speaking for myself, I thought, oh, this shows that these antibodies remove amyloid. And we now, I think, realize that that was wrong. What these first antibodies did was prevent amyloid deposition. I think a lot of people made the same mistake that I did, thinking that the antibodies caused amyloid removal. What these first antibodies did was prevent further amyloid deposition. And the penny for me only dropped really when Bart and Eric Caron produced this paper, which I loved, because it was the opposite of Monday morning quarterbacking. I always knew, by the way, the Seattle Mariners were going to win. I mean, it was obvious they were going to win. My son, who lives in Seattle, had told me that they were going to win. So that's my bit of Monday morning quarterbacking. What they said was, no. What we have to do is remove amyloid deposition. And this was a theoretical drawing that Eric Caron made, and showing what a successful trial would look like. Amyloid antibodies given there, sucking amyloid on the down, and when the amyloid deposition got down a long way and you ran the clinical trial long enough, you would see a clinical improvement. And that's shown with the solid red line versus the dotted red line of placebo. That was their theoretical side. And then these are the real slides from the actual data, showing that they fit really perfectly with what Eric and Bart had suggested. There's donanemab, there's lecanemab. These are the successful trials. Perhaps most interesting are the aducanumab trials. There are 2 aducanumab trials. I'm sure everybody certainly all the neurologists in the audience will remember the controversy about the fact that aducanumab was approved, yet only one of the 2 trials had shown statistically significant results. And sure enough, that's the lower of the 2 aducanumab trials. So I think that fits very well. And then there's the gantenerumab trial, which was released at the same time, and gantenerumab failed. So really, I think this is really the Distrupa and Karen are really, in paper, really give you a roadmap for the future. So this is the lecanemab data. I'm not showing donanemab, but the only reason I'm not showing donanemab is because it's almost exactly the same. On the left-hand side, you're seeing the amyloid removal. On the right-hand side, you're seeing the clinical effect, and you see a clear differentiation. 2 things. It clearly works. I think that arguing it doesn't work is really boring, actually, because it clearly works. But the other very important point is it doesn't stop disease. And that also is a very important point. We need to do better. So treatment has a slight benefit. Recruitment was at an early stage. I've just said this slide I've altered in a way that's not completely comprehensible now. And I'm going to say that some of the early trials had a very poor diagnostic accuracy. That isn't true of these trials because they were a biomarker validated. The original administration was intravenous every 2 weeks. And of course, there was the problem was ARIA, amyloid-related imaging abnormality. And that is caused. And there's a very good review from Greenberg on the explanation of ARIA here, which I show. What are the problems? Well, the ARIA is usually asymptomatic, but it can be serious. And if someone is taking anticoagulants, it can be very serious. And the other problem, of course, is the expense of the drug and the expense of monitoring. I should say what's happened in the UK is that these drugs both have now been approved, but they're not, this is a really unusual situation. They've not been approved for reimbursement. That is a very bizarre situation. I think it's almost unprecedented in the UK, where they pass the approval for administration, but don't pass the approval for a reimbursement. And I should say that it's led to a bizarre outcome, which there is a lot of fighting about in the UK, and that is that wealthy people pay for the administration, okay? But if they have complications, the NHS, which pays for the complications, and that is more than half the cost. So we have this bizarre situation, which in the socialist nirvana of the United Kingdom means that the rich are getting their expensive therapies subsidized by the poor who don't have access to the same therapies. You can imagine this is causing a little bit of I don't know if politicians have got souls, but it's causing a little bit of soul searching, let's say. So that's where we're up to. Now, these are the outstanding questions. Would earlier treatment result in a better outcome and less ARIA? The two things are related. I should say that when Nick Fox, my colleague, sees this, he says, what's the disease you know of where earlier diagnosis does not lead to a better outcome? So he thinks, in a sense, this is a trivial thing, but it is true, of course. And would it lead to less ARIA? Could we get treatment before ARIA? If so, is earlier diagnosis possible? And that's going to be very much dependent upon the healthcare system. I think those individuals who come to Queen Square might get earlier diagnoses. But actually, in the majority, for example, of the UK, and I'm sure this is true, and I know that this is true in, for example, in rural Idaho, those individuals never really get a diagnosis of Alzheimer's disease. But what they do, if they're lucky, is they get a diagnosis of dementia. So this is really a problem of delivery. Really, it's a massive problem for delivery. It's a problem outside the best centers here in the US, and it's a problem in most European countries outside the specialist centers. And what are the questions we need to understand? What is the underlying pathogenesis of residual decline? And the other point I think we need to understand is to what extent are the pathologies dependent or independent of each other? I showed you, mouse data showing that tangles are at least in part dependent upon amyloid deposition, but only necessarily in part. It is, individuals get tangle formation in their late age in the absence of amyloid deposition. And one can imagine, because one knows about templating, that once the tangle pathology is lit, it can become self-sustaining. So there's that. Can we use genetics as a prediction for sporadic Alzheimer's disease? This is Valentina Escott Price's area under the curve using GWAS to predict, and you see we do quite well. But let me say, these are perfect Alzheimer cases. What happened in this series is I personally went through the path reports from all the US Alzheimer centers, and if someone said there was a lot of Lewy bodies, that they did not make the cut. If they said they had vascular problems, they did not make the cut. So this is very much, the best of the best in terms of the Alzheimer centers. The other problem with this is that these are actually, I think, all white Anglo-Saxons. So although this looks pretty good, it's deficient in those two ways. It's really not real world data, it's perfect data, and it's perfect white data. We've got a long way to go. And can we use biomarkers? This is just a title from one of Henrik Zetterberg's slides. Biomarkers, of course, p-tau217, I'm sure you guys are using it here, is great as an amyloid marker, but it's great in terms of frank Alzheimer's versus frank controls. Again, it's very much the perfect situation. Is it good for very early AD against, controls? That is much less clear. So that's deeming, can we get early diagnosis? We're all very excited about trontinemab, of course. This is gantenerumab repackaged with the active site added onto the transferrin receptor, showing, and it's only gone to phase 2, so one has to be a bit careful, but showing that you get the active component shuttled across the blood-brain barrier. let me just say that about 9 or 10 years ago, not for Roche actually, for another company, I was asked to evaluate whether brain shuttles would work, and I said, not a chance. So when you get, reviews which turn out to be wrong, it's not necessarily, some conspiracy. It's just that your reviewer is wrong. And I've been wrong, certainly with this. I thought it would never work. I also thought that antisense would never work as well. So, confessions. So this is the trontinemab phase 2 data showing how quickly it works. And then this is, I'm sure you guys, certainly the neurologists have seen, that, we all see the drug company data and we all think, yeah, but that's drug company data. How does it work in practice? This is the first real world publication. And I think this is really great that it is working. I know the Mayo have got similar data. I haven't seen their publications, but I have seen it presented. And Ron Peterson explained how they do this at Mayo. Well, they might have changed, but how they did it at Mayo. They'd have a consensus meeting for each case. Can this case be put on lecanemab? And it would be both 2 neurologists, the radiologist, a neuropsych, and a psychologist, at least. So they'd have a conference about it. And then I think it was about half the patients went forward to get the drug. Well, very rigorous. Let me just say that would not work in the NHS. And, that is the problem in terms of rollout. When the Mayo Clinic really put, and I'm sure other top centers put their shoulders to the wheel, they can get these not trials anymore, get these patients through. But in the UK and in a public system, it's really not easy to see. So we have therapies work. We largely understand why previous therapies, they aren't perfect. ARIA is a problem. Incomplete slowing is a problem. But I think we're going to get better every year from now. After the Wright brothers got the first plane off the beach in North Carolina, progress was very fast. And I think that's where we are. We've done that figure of 8. Was it Oliver or Wilbur? Has landed, and now everybody knows what they need to do. And so, I think that we don't need to argue about is amyloid a good therapy or not. We just need to take the approach we know to some extent works and make it better. Of course, we should be looking for other therapies, because I think anybody who suffers from a complex disease, and I'm a type 2 diabetic, knows that where you end up is polypharmacy. And that's where I'm sure we're going to end up in Alzheimer's disease. So thank you very much, and thanks for inviting me to the Sun.
John, talking about early intervention prevention and lipids and so forth, could you comment about ApoE, which has been around? It's this mystery gene. E4 is really bad, and yet there's no ApoE-related therapy, whether direct or indirect or accidental. Do you think it is a legitimate target or not at all?
Well, I do think I definitely think it's a legitimate target. And it's of course, though, a very, very, very difficult target. It's got such pleiotropic effects. It's very, very difficult to know exactly what one should do. And so I've got no real insights, except that, like you, I'm not surprised, because I can see it's a difficult target. But if you're disappointed that there has been no successful therapy based on ApoE. I think that now we have got other lipid targets that, the good thing about having more than one target in lipid pathways is it gives you a direction of the pathway. When you've just got one point, ApoE, and it has many, many effects, it's difficult to know which of the bits of biology that ApoE is involved in is crucial. Whereas when you've got other parts of the target, other parts, you start to see pathways maybe, and it starts to be at least easier to think of ways to intervene. But I've got no special knowledge, and if I did, I'd charge you, Doug.
John, thank you for this wonderful Shiley lecture and for taking us through much of the history and providing a comprehensive view. Question about the significance of amyloidopathy and its ability to predict whether people will or will not develop clinical symptoms in their lifetime. We've been involved through the International Working Group in some controversy about what the significance is within the stratification of biomarkers, and particularly with some concern about telling people who are amyloid positive alone that they've got Alzheimer's disease and the implications for that. Do you want to comment on that issue?
Yeah, but again, I recognize it as a difficult issue, of course. I mean, of course, I had a friend of mine phone me up and say, I'm amyloid positive. What should I do? And of course, I got no answer apart from platitudes. We don't know how long people can be amyloid positive for without developing symptoms. I think it's really but that from a mechanistic point of view, you can have high cholesterol for many years before, and you might never get a heart attack. So it is it's not an entirely different situation. But that's also not really a useful answer to somebody who gets on the phone with you. So I don't really know. One of the things I meant to say, and this gives me the opportunity to say, is that the interferon pathway is not important in terms of whether you get COVID. It's not important in whether or not you COVID. But OAS and the other aspects of the interferon pathway are important in terms of whether you get bad COVID. And it's interesting that OAS and the interferon pathway is showing up again in what one might think of as bad Alzheimer's disease. So I think that maybe the interferon pathway is part of that issue. Let's see. We haven't finished that work, and other people are obviously looking too. But I can't answer your question. I'm just blathering now.
Thank you again. That was very interesting. And I was interested in, again, ApoE and what your thoughts might be on why a woman with ApoE4 carriership is at advanced risk of developing more tau than a man with ApoE4, and whether or not that plays into your work with GWAS and other work.
It doesn't, and we and others more than us, have done sex-specific GWAS, and there's very little difference. But one of the things that we did years ago is look at the gender-specific gene expression, and in fact, the immune system is more pumped up in women than it is in men. If you look at a very large number of immune genes, they have a higher expression in women than in men. And when we wrote that paper, which is now about 8 years ago, we made the point that immune genes, immune diseases, and at that time we thought of MS, has a higher prevalence in women than in men. So I do wonder if it is something, if this gender specificity, partial gender specificity, is is dependent upon the immune system. So an answer, not necessarily right, but an answer.
That's great. Thank you so much for this fantastic talk. And I want to probe that interferon story a little bit more. Just recently out of Yale, there was a discussion about the common cold. And if you mute the interferon response, even in the nasal tissue, you'll get a much worse response, immune response. It's fascinating, actually. So I wanted to see which direction you think the interferon is playing. Is the muted interferon response a bad thing, or do you think that if there's worse of an interferon response, do you want more or less of an interferon response to mitigate your AD pathology?
I think you want more of an interferon response.
Yep.
I think you want more of an interferon response. I'm really influenced by the COVID. I mean, of course, the common cold and COVID are very closely related, of course. So, I think you want more.
Yep.
I think
and that would match. That would match. Yep. So that's fascinating. It's going to be a great story and tie a lot of things together.
Hello. Yeah, I loved the historical context. Thank you so much for that amazing talk. So I had kind of two quick questions. So the first was, I was wondering, in terms of sporadic Alzheimer's, there's these protective alleles that have popped up, and there's so few of them. That's just, I only know of the APP, the one that was found in Iceland. I was wondering if any of those popped up in any of your work, and if you can see, in other populations that are also resistant to Alzheimer's, do we see the APP?
Yeah. Well, of course, the clearest example is ApoE2. In terms of protective, it's often forgotten, but that's the elephant in the room, if you like, in terms of protective.
Yeah, I was talking about mutations, like.
Sure, no, I understand, and so we've looked and we've never found any. When I was at Mayo, we searched for ApoE variants, and we found, actually, and the Mayo group have found, I think, 2 or 3 more variants which are different in their risk from ApoE3 and from ApoE4. So just like just like the
no APP in any of.
No, I've never found. We have never. Well, we have found APP mutations, but
protective.
No, we have never found. We have never found. But we have found the what's called the Icelandic mutation in the Finnish population. So we have, if you like, confirmed that, but not found novel ones.
I was also intrigued by your description of amyloid A-beta and how it sticks to lipids and the secretion. And it made me think, of course, everything you're saying kind of makes sense from a neuropathologic perspective too, because you see these, dead nuclei in the center of the plaques and things like that. But clearly A-beta is secreted from neurons. There's no question about that, right? Because we can do ELISAs from extracellular. So what do you think about that?
Yeah.
Because that goes against your hypothesis.
That's what I think about that. Yes, everybody says that. They say, but, I wonder if it's free or if it's in some sort of conjugate, with the lipid still.
It's definitely outside the cell, though.
Yeah. No, I absolutely, of course. I'm not answering your question. I realize that. But believe me, I've thought of that, and others have questioned me before.
Thank you so much. Yeah.
And I think that's an excellent place to stop this wonderful discussion. But we really want to thank you, Sir John, for doing this work. And thank you for really enlightening us through the Shyly Endowed lecture. Thank you.
Thank you very much.
Thanks so much. Appreciate it.